We are taught from a very young age to view bacteria as the enemy. We scrub our hands, sanitize our countertops, and take antibiotics to eradicate the invisible threats lurking all around us. It is a completely logical habit. Public health messaging rightfully focuses on the microbes that cause disease, but constantly treating the microscopic world as a hostile battleground blinds us to how life actually works at that scale. We have to question our first assumptions. This visualization maps out microbial populations. That tiny sliver represents the harmful pathogens, like the strains of E. coli that cause food poisoning. The massive expanse surrounding it represents the billions of microbes that are either entirely neutral or actively keeping us alive. To understand this, think of a microbe less like an invader and more like a roommate. In a biological relationship known as symbiosis, this roommate pays rent by providing services you cannot perform yourself, like synthesizing essential vitamins. In this microscopic view, you are watching yeast, a single-celled fungus, rapidly expanding and aerating dough to make bread. Inside your own body, populations of gut bacteria are currently breaking down complex carbohydrates that your human cells are simply incapable of digesting. Even a beneficial roommate becomes a threat in the wrong environment. A gut bacterium that aids digestion can trigger a lethal immune response if it migrates into the sterile environment of the bloodstream. Danger isn't a fixed identity. It's a product of the relationship between the microbe and its location. Once we understand what microbes actually do, we need to audit our assumptions about how they are built. Take the term cell wall. Language matters, and the word wall immediately brings to mind a solid, impenetrable brick barricade designed to isolate whatever is inside from the outside world. This cross-section illustrates the true function of a cell wall. In plants and bacteria, the wall provides rigid structural shape to prevent the cell from collapsing. However, it is highly porous. Essential nutrients and water are constantly passing right through it. If we peel away that rigid outer layer, we find the fluid cell membrane underneath. Animal cells lack a cell wall entirely, relying exclusively on this flexible, moving membrane to act as their boundary. Let's pause for a quick assessment. True or false? A cell wall stops all substances from entering the cell to protect it. The answer is false. A solid barrier would starve the cell. Permeability is a requirement for survival. Cellular boundaries evolved to regulate the exchange of chemicals with the environment. They were never meant for absolute isolation. Moving inside the cell, we encounter another pervasive myth: the idea that every living cell possesses a neatly defined membrane-bound nucleus containing its DNA. This side-by-side comparison shows the reality. While plant and animal cells secure their DNA inside a distinct nucleus, bacteria operate differently. They use a nucleoid, an unspooled, free-floating region of genetic material completely lacking a containment membrane. We see a similar variation with vacuoles, which are frequently taught as universal cellular storage units. Watch how this structure changes across biological kingdoms. A plant cell requires a massive central vacuole to maintain internal pressure and structural firmness. Animal cells only utilize tiny, temporary storage vesicles, and in bacteria, the structure is entirely absent. Internal organelles are highly specialized adaptations tailored to specific environmental pressures. They are not a universal checklist that all forms of life must follow. Much of our misunderstanding stems from the tools we use to observe cells. It is tempting to believe a microscope can simply zoom in infinitely if we just add thicker glass. To understand why that fails, we have to separate magnification from resolution. Magnification simply makes an image appear larger. Resolution is the ability to actually distinguish two separate points of fine detail. This photograph shows the standard light microscopes found in classrooms and early laboratories. Due to the physical wavelength of visible light, these instruments hit an absolute physical limit. They max out around 1,000 times magnification and cannot resolve any object smaller than 0.2 micrometers. If you try to magnify an image beyond that physical resolution limit, you do not uncover new details. You only get larger, blurrier shapes, which historically led to false assumptions about what was actually happening inside a cell. Human observation is strictly bound by the physics of light. For centuries, that limitation blinded us to an entire foundational layer of the microscopic world. Lying entirely below that resolution limit is the virus. Measuring between 20 and 300 nanometers, Viruses are too small to interact with visible light in a way that our standard microscopes can detect. This geometric viral particle breaks every fundamental rule of life. It has no cell membrane, no cytoplasm, and completely lacks internal organelles. Because a virus lacks the cellular machinery to process energy or reproduce on its own, it exists as an obligate intracellular parasite. It relies entirely on hijacking a living host cell to survive. This cyclical flowchart maps out the rapid viral life cycle inside a host. The virus attaches to the host membrane, enters the cell, forces the host's own organelles to replicate the viral genetic material, and finally triggers an explosive release to infect neighboring cells. Let's do one final assessment pause. True or false? You can easily see the viruses causing your cold using your classroom light microscope. That is false. To see a virus, we have to bypass light waves entirely. This electron microscope uses high-speed electron beams to achieve a resolution thousands of times sharper than a traditional lens, finally bringing nanometer-scale particles into focus. The microscopic world rarely fits into neat human categories. Identifying the limits of our tools and the precise behaviors of these biological rule breakers is what allows us to distinguish a beneficial symbiont from a hidden pathogen.